Hello, my name is Aureliano Stingi, and together with Luca Cirillo we wrote a review here on BioEssays in which we evaluate the oncogenic potential of the SARS-CoV-2 infection. Since the first outbreak in 2019, more than 160 million people got infected by SARS-CoV-2. And besides the severe symptoms, there is rising concern about the long-term effect of COVID-19. Here we review evidence pro and against a possible role of SARS-CoV-2 in cancer onset. Cancer is a multifactorial disease with several causes, and one possible cause is the viral infection. One infamous example is the HPV, the human papillomavirus. Some HPV strains bear an oncogenic potential. In fact, HPV is able to encode for two proteins, E6 and E7, and those two proteins are able to inhibit two important oncosuppressors like p53 and the retinoblastoma protein, RB. p53 and RB control the cell cycle progression, and they are two important checkpoint proteins. Their inactivation often leads to cancer. One HPV feature that makes it oncogenic is the ability to cause chronic infection. The long permanence is also causative for the cancer onset. But now let's give a look to SARS-CoV-2. We hypothesized that SARS-CoV-2 may bear an oncogenic potential because SARS-CoV-2 present two proteins that are highly conserved with SARS-CoV-1. SARS-CoV-1 is the responsible for the 2009 SARS pandemic. Those two proteins are NSP15 and NSP3, and those two proteins has been shown to interact and also to inhibit at different level RB and p53. So therefore we speculate that also SARS-CoV-2 may bear an oncogenic potential like SARS-CoV-1. But differently from HPV, the SARS-CoV-2 infection is an acute infection that resolves itself in a couple of days. So the permanence of the virus inside our body is quite limited. So on one end, the oncogenic potential of SARS-CoV-2 infection is supported by the fact that SARS-CoV-2 contains two proteins that are highly conserved with SARS-CoV-1, and those two proteins are NSP15 and NSP3. Those two proteins are indicated to be able to interact and also inhibit p53 and RB. But on the other hand, we have an infection that is an acute infection that normally resolves with cell death or cell cycle arrest because it's characterized by high cytotoxicity. And to resolve this conflict, we proposed a series of experiments with an experimental roadmap. So first of all, histology will clarify the presence and the abundance of NSP15 and NSP3 in the tissue of the infected patients post-mortem. Second, epidemiological studies potentially will show a correlation between the recovered from the SARS-CoV-2 infection and the presence of the emergence of some type of cancer. And third, animal models and organoids will help to clarify the biology and the molecular biology behind our hypothesis.